Hi there, everybody. My name is Sarah Trenfield, and I am a Strategic Stakeholder Engagement Manager here at the Medical Research Council, the MRC. I've been leading this year's MRC Millennium Medal 2020 competition, which is the MRC's most prestigious prize awarded annually to a researcher that demonstrates scientific excellence and impact. I'm delighted today to be joined by Professor Sir Rory Collins, who in December 2020 was announced by MRC Council as this year's MRC Millennium Medal 2020 winner. So Rory is a British Heart Foundation Professor of Medicine and Epidemiology at the University of Oxford. He's also the head of the Nuffield Department of Population Health and a Fellow of St John's College, Oxford. So Rory is also the Principal Investigator and Chief Executive of UK Biobank, which is a large-scale biomedical database and research resource that can be accessed globally by approved researchers. So firstly, I would like to offer a very warm welcome to Sir Rory. Thank you very much for doing this interview today, and a very big congratulations again on being awarded this year's MRC Millennium Medal.
Thanks very much.
So can you tell us, how did you feel when you found out that you'd actually won the medal?
I felt that I've been incredibly lucky. I mean, I know the old adage that you make your luck, but also you do need to be lucky. And in particular, I think as a young doctor, I was very lucky in getting to meet Richard Peto, who then taught me how to do research and also what mattered in research and what kind of research mattered. So yes, you have to work hard, but I think you also need to be lucky.
Yeah, but I think, as you say, you do make your own luck, and I think a number of your outstanding achievements, I could think, are testament to that. So can you tell us a little bit about your research, just a bit of background into some of the work that you've done over the past number of years?
Well, I originally trained in medicine and then in statistics, and I was very interested in the idea of combining mathematics, statistics, and medicine. And in particular, as a young doctor, I really fell in love with the idea of the randomized controlled trial. It was invented by the Medical Research Council. The first randomized controlled trial in humans was done in the 1950s with the drug streptomycin for tuberculosis. And I just feel that it's an incredible invention in order to understand what treatments work, what treatments don't work, what treatments are safe and what treatments are not. And that was really what got me into research. The idea that there was this extraordinary tool that one could use, and particularly use for large populations of patients to work out how to treat them better, treat them safer. And that's really what seduced me into research.
Yeah, that's I think it's something that's very timely as well. I think with the COVID-19 situation, I think actually the general public are being much more informed about what a kind of a randomized control trial is and how that can actually impact and really change, I guess, the world in terms of medical research. So I think going into kind of some of the randomized control trials that you have coordinated in the past. So I think one of your major achievements is that you coordinated a trial around statins. So actually this work has kind of saved the lives of millions of patients around the world, because one of your outcomes is that you found statins, which are now almost available as low-cost treatments, are actually effective at preventing severe cardiac events, including heart attacks and strokes. So I'd just be really interested to almost know a little bit more about this work. So how did you come up with this idea of even testing statins, and how did you find out that they were effective against these conditions?
I think what was important was that we found that lowering cholesterol was much more important than people had recognized. And it was a beautiful combination of observational epidemiology and randomized controlled trials. So when people looked at the association of heart attacks in particular with cholesterol levels, there was what we would call a curvilinear association. So as the cholesterol got higher, the curve took off. It seemed to go almost vertically. And so people interpreted that as saying, well, high cholesterol is bad. But I mean, again, in the year of COVID, everybody has got used to the idea of exponential curves and log curves.
Yeah.
And if you take those same data and plot them on an exponential, a log plot, then what you actually get is not a curvilinear, where it seems to be going up only in people who have high cholesterol, but you get a straight sloping line, which suggests that the same absolute reduction in cholesterol is associated with the same proportional reduction risk throughout the cholesterol range. That is, that for particularly Western populations and well into Asian populations as well, lower cholesterol is associated with lower risk. That was quite a revolutionary idea, and it was merely changing the scale on a graph. And that's what we set out to test in the heart protection study, which was funded by the Medical Research Council and the British Heart Foundation, to see whether in people who are at high risk of having a heart attack or having a stroke, lowering their cholesterol would lower their risk, irrespective of whether it was considered to be high or not. And that trial showed that that was indeed the case, that in these high-risk individuals, what mattered was their risk, not their cholesterol level. And irrespective of their cholesterol level, lowering that cholesterol lowered their risk. Each 1 unit, 1 millimole, lowered their risk by about 1/4. And if you lowered it by 2 millimoles, you lowered it by 2/4 roughly. So that was really transformational in the way in which we all think about cholesterol. So instead of focusing just on people with high cholesterol, we completely shifted to the way clinicians do it now, which is to focus on people at high risk and to lower their risk by giving them drugs, the statins, that lower their cholesterol. And we'd observed this association in the epidemiology, and then the statin drugs were started to be developed, actually by a Japanese chemist who found them in a fungus initially, and then they of course created them synthetically. But pharmaceutical companies developed these drugs, and we worked with them to assess their early safety and efficacy in terms of lowering cholesterol. And then, as I say, with Medical Research Council and the British Heart Foundation funding, and indeed the companies, we were able to do this very large-scale trial across the UK that demonstrated benefits, and benefits for a very wide range of different types of people: older people, younger people, women as well as men, and also, much to many people's surprise, not only preventing heart attacks, but providing the first definitive evidence that you could lower the risk of strokes due to clots as well.
Wow, it's truly incredible, and I think the scale of that impact is just massive. So, do you know how widely statins are used nowadays? Are they used kind of globally?
Oh, in the UK alone, millions of people. So worldwide, we're talking tens, perhaps, I don't know, hundreds of millions of people at high risk, and a much wider range of people using them. And of course, initially, they were patented and so quite expensive, but they've now been around for long enough that they've all pretty much come out of patent, and so their cost falls enormously. In the UK, we're talking a few pence per day to take very effective drugs that can lower your cholesterol by about 50% with a small tablet. And the other thing that the trial showed is how well tolerated and safe they are. The beauty of a randomized controlled trial, particularly if you use a dummy tablet to blind whether or not people are getting the real thing or a dummy tablet, what we find is that there's very little difference in any symptoms of side effects in people who are taking the real thing versus the dummy tablets, which I think is, again, very reassuring. And with the MRC support, what we've then done is work with investigators around the world who've done other randomized trials of statin therapy. They have provided us with all of their individual patient data, and we've then been combining those data in order to produce even more information, more reliable information about the efficacy and safety in different types of patient. And so, again, none of that work would have emerged without the Medical Research Council's long-term funding for that project.
Brilliant. And I think also in the 1980s, you kind of coordinated another, almost a mega trial, they're so-called, the ISIS-2 mega trials. And this was coordinated in around 130,000 patients, and you essentially were able to have findings which led to improvements in the emergency treatment of heart attacks. So are you able to tell us a little bit more about these trials and how they have also changed medical practice today?
Yes, so the Medical Research Council invented the randomized trial in 1950. In a way, it was a little bit of a trick by Austin Bradford Hill, in that he was a statistician, ran one of the MRC units, and he thought for a long time that this concept of randomizing people to getting a treatment or not was going to be critical for the reliable evaluation of treatment. But he thought at that time the medical profession would consider it unethical for him to propose it. So he waited for his opportunity, and his opportunity came with streptomycin, which was very limited in supply. And he proposed in an MRC meeting, why don't we make it available through a lottery? Because we don't have enough for everybody, we'll just do it by a lottery. But of course, he essentially introduced randomization by doing so. In that case, streptomycin was incredibly effective. So in some respects, a randomized trial wasn't needed because the benefit would have been so obvious. But it did introduce the whole concept. Unfortunately, I think what it also did was it made people think that you could do small randomized trials and get clear answers. And unfortunately, most treatments are not streptomycin. Most treatments have moderate benefits. And so you do need to not only randomize to avoid bias, but you need to include very large numbers of patients in order that you reduce random error, so that both your biases and your random errors are small by comparison with the effect you're trying to pick up. And that was really where I think the reinvention of randomized trials by Richard Peto was so critical in the early 1980s. When I joined his group in Oxford, he was making the case very strongly that there had been a failure of randomized trials to really detect worthwhile benefits because they had been too small. And so in collaboration with the cardiologists in Oxford, they were planning to do a very large, very simple randomized trial of the treatment of heart attack, emergency treatment of heart attack. That was when I turned up in his office, and he was looking for somebody to help make this trial work, and he offered me a job. And so I joined him to learn on that trial. And with him and with a colleague, Salim Yusuf, we looked at all the previous trials that had been done in heart attacks, emergency treatment of heart attacks. And we found that there had been a lot of trials done with a clot-dissolving treatment called streptokinase. It's an enzyme produced by the bacteria in your throat. And it was made by a company in Germany called Boehringer. But really it wasn't being used medically because there had been a lot of small trials for the emergency treatment of heart attack, none of which had been big enough to demonstrate whether or not it was beneficial. And it does cause bleeding. So they could see the bleeding, they could see the potential harm, but they couldn't see any benefits. So it had largely been abandoned. But when we put together all the evidence from these trials, it looked to us as if there might be a reduction in mortality by about a quarter, but it would need a big trial to demonstrate that. And so we got agreement to do that. We got the drug from the company and a bit of money. And we ran this very simple randomized controlled trial of streptokinase. And in fact, at a lunch one day, we were thinking, well, if we're going to do this trial, what else might we test? We thought, well, we're going to give a drug to dissolve the clot in the coronary artery, or that's what we hope we're doing. why don't we try to prevent the clot from reforming? And we'd done work with aspirin, and we thought, well, what about giving people half an aspirin, a baby aspirin a day for a month, and seeing whether that is beneficial. So we went forward with that trial, with the so-called ISIS-2, the International Studies of Infarct Survival, myocardial infarction, heart attack, and the ISIS is the river that goes through Oxford. And we had been building up a collaboration in the UK with dozens of hospitals across the UK and beyond in many countries in Scandinavia, Australia, etc., and North America. And we got people in those hospitals to take part. It was very, very simple. Patient came in with a heart attack. At that time, if you had a heart attack, what happened was you were given some pain relief, and then they put an ECG monitor on you and they watched you. There was no treatment. So we said, you just ring up and we will tell you at random which treatment pack to give. And they would put up a drip and give an infusion for an hour of either streptokinase or a dummy infusion. And then they would give this small baby aspirin to the patient or a dummy tablet for 1 month. The streptokinase reduced mortality by a quarter. And then we looked, and the aspirin reduced mortality by a quarter. And the combination halved mortality. So we'd gone from nothing working to these incredibly simple treatments that were generic, producing a big benefit, an extraordinary benefit in on mortality. And they were shown, because the trial was very big, it was 17,000 patients, they were shown to be safe. And so very rapidly, there was a transformation in the way in which heart attack patients were treated around the world. And it then went on to look at better ways of dissolving the clot with newer agents, better ways of adding things to aspirin, and then finally to looking at mechanical ways of opening up the coronary artery with a tube passed up into the artery and blowing up a balloon, and then maybe even leaving a little bit of metal there to hold the artery open. But it all really followed from looking at these old small trials that suggested there might be a benefit, but then doing a large trial, which was very, very simple, very, very low cost, very, very fast, and demonstrating this benefit. And that really introduced, I think, a shift in many people's views about not only randomizing, but needing to do much bigger trials.
Yeah, so this is what I was going to ask as a follow-up, really, was kind of have trials kind of expanded in size in terms of patient numbers now? Like, is it more common now to do these large, almost mega trials, or are there still some kind of a series of small trials going on, or something in the middle?
Well, for a while, things were really very good. People understood the need to randomize now. They understood the need to do very large trials. And we saw a real explosion in answers coming out in cardiovascular disease and in other conditions as well. But unfortunately, what's then happened is increasing regulation and bureaucracy, which was intended to make trials better, has actually made it very much harder and very much more costly to do randomized trials. And so over the last 20 years or so, we've seen major obstacles to the evaluation of treatment, inadvertently caused by rules and regulations that were intended to do good. The EU Clinical Trial Directive was put in place in 2001. By 2009, the European Union had done a review of it and found that it had failed in its aims, and had made it much more complex and costly to evaluate treatments to the detriment of patients, was their own conclusion. So I think now it's really important to look at how we streamline those processes. And there are very few things that one can say that positively about 2020. But I think the one thing we have seen is, as you mentioned, the power of the randomized trial. And in particular in the UK, the power of this concept of very streamlined randomized controlled trials. And when we look at the UK recovery trial, which was the trial that demonstrated that dexamethasone reduced mortality by 30%, particularly in the patients who are being ventilated. That was the ISIS-2 trial repeated. It was pretty much exactly the same design. And in fact, Martin Landry, working with Peter Horby, had the ISIS-2 protocol open on his desk, along with the forms, as he was writing the protocol for the recovery study. And it had input from Richard Peto, who's now emeritus, on how to keep it streamlined. And indeed, the same design approach was adopted by the WHO for their parallel solidarity trial. So I think there's a real opportunity now to, if you like, go back to the future of really taking advantage of the randomized controlled trial, do them larger, do them more streamlined, and ensure that the guidance is actually supportive of generating reliable evidence rather than inadvertently causing unnecessary obstacles. So I'm excited about the reinvention of randomized trials for the 21st century.
That's absolutely amazing. And just on a side point, how does that make you feel, kind of knowing that, you know, the research that you've been doing over X number of years is having, you know, has had and is still having such amazing impact in terms of both for cardiovascular disease, but also for, as you mentioned, these latest COVID recovery trials? How does that make you feel, almost as a researcher, as a person?
Well, I mean, the thing that matters, you know, the thing I was taught by Richard at the beginning that matters is what's going to affect the largest number of people. And so I think the beauty of the result with dexamethasone is not only is it a very definite finding, but it's cheap and available, accessible to people around the world. And I feel the same thing about the ISIS-2 trial of streptokinase and aspirin. Although it has led on to improvements in the way in which one opens arteries and the way in which one keeps them open for high-income countries. So if you or I have a heart attack in the UK, we will get rushed into hospital. We will get into a theatre where they will do primary angioplasty. They will put a balloon up into our artery and open it up and put a stent in. But still, despite the ISIS-2 results having been published in the Lancet in 1988, the vast majority of people who have heart attacks around the world will be getting a thrombolytic agent, streptokinase or one of the newer agents, generic drug, and they'll be getting an aspirin. So it identified ways in which one could do better, but it also identified ways in which one could do pretty damn well for the vast majority of the world's population. And so I feel that about what we did in 1988, and I feel the same about what Peter Horby, Martin Landry, and the whole of the recovery team, and most importantly, all the doctors around the UK who took part in that trial, and of course patients. Recovery trial allowed doctors on the front line to be involved in research that makes a difference. And I think that's another really positive aspect about it. To some extent, the guidance and the rules and the regulations have disenfranchised a lot of junior doctors from being involved in these trials in the way they were with the ISIS acute heart attack trials, and the way that they have been now with the recovery trial. And my experience is once you've been involved in one trial that gets a really clear answer, it's incredible, you're hooked. It's very, very difficult to not want to see more results like that.
Yeah, I think you touched on a good point that I think, you know, we've talked about it initially, but kind of public interest in science and participation in research studies has really increased because of COVID-19. So I guess what lessons do you think we can take from this going forward? Because, you know, there's high participation at the moment, but how can we encourage high participation going forward as well in different studies and in different situations?
Well, as you say, with COVID, what the public has seen, and you know, more than just the public, what government has seen, is that medical science can make a really big difference. I mean, we're going to get out of this because of medical science. And it's not something you can just turn on and turn off. So, the power of the Medical Research Council is it's there all the time. You have power that you can access rapidly. I mean, our department has a Medical Research Council unit. We don't do any infectious disease research, but within a few weeks, we became an infectious disease research group because you could apply that power to something else that was really important. And I think, I hope that by continuing to demonstrate the power of medical science and by continuing to take advantage of what we've learned, so how you can streamline the studies that are done, how you can streamline the processes so they can be done more quickly and more efficiently, how you can involve more people, more doctors around the country by making it simple for them to do so, and more patients involved in the trials. You actually make it available to them rather than something that only occurs for a minority of people. Then it becomes part of normal practice. And people talk about a learning health system. But I think that 2020 has demonstrated that probably the UK is in a unique position to have a learning healthcare system. We have healthcare records that allow us to do research that most other countries, unless they're relatively small, just can't do. And we have a research community supported by, amongst other, the MRC, but again, charities as well, that can really do extraordinary research. So I think working together, the NHS, the research funders, and the researchers, along with the public, something very, very good can come out of something that was very, very bad, COVID-19.
So, kind of more towards some of your work on UK Biobank, if that's okay. So, I think one of the reasons for you being awarded this year's MRC Millennium Medal was due to your visionary leadership of UK Biobank. Can you tell us a little bit more about UK Biobank? So, in terms of its aims, its participants, and also the world famous data sharing model.
Well, the vision for UK Biobank came from the Medical Research Council and the Wellcome Trust. They, ahead of anyone else anywhere in the world, saw the importance of setting up a very large-scale prospective study. And they put their money into it. I think that was an incredibly courageous decision because, frankly, it was never going to produce anything useful in the first 10 years or so. It was going to be a lot of money to set it up at the scale that was needed. UK Biobank, 500,000 men and women from around the UK, aged 40 to 69 when they were recruited between 2006 and 2010. But then they were going to have to be followed up through health record systems to find out what health outcomes occurred. And until they did develop health outcomes, it was a lot of money, a lot of work, but not very much in terms of output. And the MRC and the Wellcome Trust knew that, and they put a big bet down. No one else did that. What it means is that because of 500,000 altruistic people from around the UK, because of the vision of the MRC and the Wellcome Trust, the UK has established, I think, a unique resource for global research. It had a lot of funding to get the 500,000 people in the first place, but it has had additional funding, again, from MRC, Wellcome Trust, the NIHR, and the charities like CRUK and British Heart Foundation to enhance the resource so we know even more about the participants. We have genetic information, first genotype, now exome sequence data, so the sequencing of the 3% or so of the genome that actually produces a protein. Now ongoing whole genome sequencing on the 500,000 people. When you think whole genome sequencing on one person was done only about 20 years ago, now we're doing it on 500,000 people as if it were routine. It will be the biggest genetic study anywhere in the world. We have imaging, again funded by the MRC and the Wellcome Trust, on 50,000 of a planned 100,000 people. And now we're getting a lot more external investment in it from pharmaceutical companies for assays and other measures. So they had the vision, the MRC and the Wellcome Trust, set up a very big cohort. But I think even they, certainly me, I think was surprised by how much more is being put into it, and how fast that's occurring. So it's not just the scale of the study, but also the depth, real detail on all of these participants who are now being linked through to the UK health record systems, which are very, very detailed about people's health. And because of the model that the MRC and the Wellcome Trust had at the beginning, our job was to build it for others to use. So although I'm described as a principal investigator, I've probably done less research on UK Biobank than pretty much anybody else. My job was to get people to tell us what would make it useful, and then for us to develop it and make it available. And there are now thousands of researchers around the world using it and discovering things that they just would not have been able to discover in a smaller study or a study that was much less detailed. And it will continue to be, if you like, ahead of the game. And as time goes by, it will get more and more detailed. There'll be more and more information. It will become more and more valuable. And I think over the next 5, 10, 15 years, it will generate an increasing number of important findings for understanding why people get a particular disease, and thereby understanding ways in which to prevent and treat those diseases.
It's almost as if, it's a high risk in terms of having to spend a lot of money up front, but with very high reward. And I imagine kind of rewards for a number of different diseases. So are you able to comment on some of the, I guess, diseases that have been researched into as a result of UK Biobank?
Well, I think it's more about, at the moment, concepts that have come out of UK Biobank. The one that really springs to mind for me is the concept of the polygenic risk score. So before UK Biobank, there was a lot of argument as to whether one would be able to use genetic variants that produced modest effects on risk to identify people who were at particularly high risk. All that argument got wiped away by the ability to demonstrate that you could in UK Biobank. And initially, the finding that you could identify about 3, 4, 5% of the population who, by combining lots of genetic variants across all the genome, each of which had small effects, some good, some bad, you could identify 3 or 4 or 5% of people who had a risk which was equivalent to somebody with a genetic mutation, a single gene disorder, such as familial hypercholesterolemia. Now, familial hypercholesterolemia, if you have that gene from one of your parents, then you tend to have a heart attack in your 30s, 40s, because your cholesterol's high. If you get it from both parents, then you can start to get heart attacks in your teens because your cholesterol level is really very high. But you find people who have familial hypercholesterolemia about maybe 0.5%, but you could find maybe 5% through these polygenic risk scores that had an equivalent risk. The polygenic risk score like that also worked for things like breast cancer. So you can find a few percent of people who have a risk, a few percent of women who have a risk which is equivalent to having the kind of single gene disorders of BRCA1, BRCA2, these mutations that are strongly associated with breast cancer risk at a young age. And of course, you can do this with a very simple test, a genotyping chip, which costs maybe, I don't know what it is now, but maybe £20, which gives you all of this genetic information. So you can get a polygenic risk score for heart attack, polygenic risk score for breast cancer with the same test, polygenic risk score for colorectal cancer, et cetera, et cetera. So people have shown that you can pick up about 25% of the population that are at in that top few percent for maybe 15 or 16 different conditions. So I think the possibility that these genotype scores will be used and genotype testing will be used in the general population is quite likely, because it will be so valuable for identifying people who are at high risk before they develop disease. So when we think about breast cancer screening is done based on age, because that's the strongest risk factor. But you could use a polygenic risk score to say, well, instead of screening women from the age of 50 for breast cancer, we'll screen this individual at the age of 30 because they're in the top few percent. This individual we might not screen at all because they're in the bottom few percent. The same with statin drugs, cholesterol-lowering drugs. Largely, GPs give statin drugs on the basis that they put some stuff into the computer and it looks like magic, but mainly it's driven by age and gender. So if instead you had a polygenic risk score, which just prompted the GP to say, this patient is at very high risk because of their risk score, you should consider prescribing statin, or this patient is not. And so I think that is a close to prime time, as they say, result that could really transform the way in which we use preventive strategies within healthcare systems.
Yeah, definitely. It's almost kind of moving away as well from almost treating the symptoms as they arrive towards actually, before symptoms arise, actually knowing that that condition may occur and how can we prevent that. So it's almost changing that traditional treatment model that we have.
And of course, the UK is really well set up to do this because we have a National Health Service, we have an NHS digital with electronic health record systems, and the GP systems are incredibly sophisticated. So one can imagine ways in which the screening services would be screening on the basis of genetic risk score, not age, and sending out an invite for breast cancer screening or colorectal cancer screening, prostate cancer screening, or a prompt to the GP to say this patient should be considered for a statin, et cetera, et cetera. So it's something that could be implemented in a relatively straightforward way within the kind of system we have here. So I think the 500,000 people in the UK Biobank, that alone, I think, could make a transformational impact on the way in which healthcare system is provided within the NHS. So it benefits them and their families, but also in countries around the world.
And has the UK Biobank, has any data kind of been drawn up from UK Biobank that has aided research into COVID-19 from the genetic side of things?
Well, the beauty of UK Biobank is we have very large numbers of people data. We have all of this genetic information and a lot of other information about them in terms of various exposures. And we have all the approval to get linkage, their consent, to link into their healthcare record systems. So it was relatively straightforward, albeit a lot of work by the team, to extend the linkages to the testing data for coronavirus, to the intensive care data, and working with NHS Digital to get more frequent updates of the health outcome data than we had typically been getting. Because previously we were getting them every year or so because it was a chronic disease largely study. But because of the fast moving pace of COVID-19, we moved that up to getting things like the testing data every week or every couple of weeks, and the hospitalization and the death data, and now the primary care data much more frequently. So that researchers could look to see, are there genetic, for example, genetic or lifestyle or environmental predictors of a bad or good outcome, or getting infected or not. We then thought, well, are there other things that we could do? Because we have 500,000 very, very cooperative, supportive, altruistic participants. And early on, we were asked by Jeremy Farrar and Patrick Vallance at the Wellcome Trust and the Chief Scientific Advisor to the government, could we find out about the prevalence of infection over time? And so we wrote to UK Biobank participants and said, if we send you a finger prick device, could you take a blood sample and just mail it back to us? And we said, also, do you think you could get your children or grandchildren to agree to do this as well? We were overwhelmed. We had over 100,000 participants said they would take part. We only were looking for 20,000. So we got 20,000, and they have been, every month, we send them a kit, and every month they send us back the blood. I mean, the compliance is extraordinary. They've been doing that for the last 6 months, and we now have this very lovely data showing the seroprevalence, so seeing the prevalence increase during the first wave. And also then we can see over the months, do the antibody levels persist? And what we're seeing is that they do. So again, contrary to some of the earlier concerns that antibody levels and therefore protection rapidly falls off, the data we have is indicating, and we're doing more work on that, that protection or antibody immunity persists for at least 6 months. And then we thought, well, what else could we do? And the MRC and the Wellcome Trust had funded an imaging study, which I mentioned, where we're halfway through. We'd imaged 50,000 of 100,000 participants, and we paused it because of the pandemic. And we thought, well, rather than close it all down and lose all the staff, why don't we invite back some of the people who've been infected, who've been imaged previously, and some people who haven't been infected who've been imaged previously. Because there's been a lot of discussion about whether the coronavirus has effects on body organs, the heart and things like that. The problem for everybody who's trying to study that is they don't have the imaging measures beforehand. UK Biobank has 50,000 people who've been imaged. We've got magnetic resonance of their brain. There have been concerns about the effect of infection on the brain. We have magnetic resonance imaging of the heart, the body. We have imaging of the carotid artery before infection. And so now we're just about to launch a study with the support of the MRC and Wellcome Trust, where we're essentially repurposing our imaging centres to bring back participants who were imaged, some of whom have been infected and some of whom have not, so that we can compare, knowing what they were beforehand, we can compare whether there are differences in those who were or weren't infected afterwards, making it a unique resource for studying the effects of the coronavirus on body image measures. And then we thought, well, what else could we do? Again, with the same question of what does infection do in the longer term? We've been given by the Department of Health 500,000 self-test kits for testing for the antibody. And our plan is to send those out to participants and ask them to do self-testing and tell us, do you get a positive antibody result? If we put those data in, then researchers from around the world will be able to look to see whether having been infected versus not is associated with all kinds of different health outcomes during the years to come. We'll be able to see whether infection is associated with any kind of mental health issues, for example, or anything around fatigue or all sorts of health outcomes that could not be studied any other way. And of course, we can also see whether there are any determinants of people having a longer term effect, because we've got all this genetic and other information on the participants. So we've been looking at ways in which we can enhance the resource to give us answers quickly, like the seroprevalence study, in the next few months with the imaging study, but also in the longer term to be able to assess the so-called long COVID concept. Is that really true? How important is it? And if there are effects, what are those effects? What kinds of conditions are affected?
That sounds really exciting, and I think I'd really look forward to seeing some of the outcomes from those studies, and I'm sure they will continue to have impact around both nationally and also around the world. So aside from the UK Biobank work, do you have anything else kind of next on the pipeline? I'm sure you do.
Well, mainly going back to randomized trials. I'm very keen over the next few years that we take advantage of people's recognition of the importance of making it possible to do randomized trials more easily and engaging the public and doctors more in randomized trials. I think the UK, as I said, they invented them in the 1950s, and I think in the 2020s we have the opportunity to really reinvent them for the 21st century. I know the Wellcome Trust and the MRC are keen to move that agenda forward, and I'm very keen to work with them on that.
That's brilliant. And another question. So I'm sure that there's a number of people who have watched this interview who are perhaps young and aspiring scientists or quite early on in their career and have essentially watched this and been quite inspired to really see what you can achieve over the course of a research career. So I was just wondering, do you have any advice that you can give to those kind of young and aspiring scientists, or those who may be future MRC Millennium Medal winners?
Well, as I said at the beginning, I really do feel I've been lucky, and I was lucky in the people I was working with. And I was lucky to work with somebody like Richard Peto, but I've continued to be lucky in the people I work with. I think early on, one really does need what I would call a sponsor. I was funded by the Medical Research Council, a grant that Richard had to work on leukemia trials. So I worked on leukemia trials for the MRC while also doing trials in the emergency treatment of acute MI. So you need somebody who's going to look after you in those first few years and who believes in you even when you don't, frankly. The first few years, the first several years of a research career are one of uncertainty and anxiety. And you need someone who believes in you probably more than you believe in yourself to make it possible. Yeah, you have to work hard. Yes, you have to believe in what you're doing. But I do think that the people you work for and work with can make an enormous difference, particularly at that time.
Brilliant. Yeah, I think that's really, really sound advice. So on the 29th of March, we've got the MRC Millennium Medal Award ceremony. Very exciting, where you've kindly offered to present a lecture on some of your research. Are you able to give us a bit of a teaser as to what you're thinking of putting into your lecture? Well,
I think perhaps you've guessed from what I was saying about what I'd like to do over the next few years that I really do want to strike while this particular iron is hot on randomized trials. I think the UK is uniquely placed. I think the people in the UK, the people who are at the top of the major funding agencies, recognize the value of randomized trials and the power that the UK has potentially as an international leader in this area. When one thinks about COVID-19, the recovery trial and other trials done in the UK have shown the world what can be done, and I think we should build on that. So my my topic will be very much about that opportunity.
Brilliant. I'm looking forward to it already. So with that, I'll bring a close to the interview. Do you have any last thoughts or remarks, Sir Rory?
No other than to say thank you to the MRC for having been there and continuing to be there for me, but also for so many other researchers across the UK. One puts in program grants and project grants and things like that, but it's also that kind of core, that long-term support that makes the difference for people.
Brilliant. So thank you so much, Professor Sir Rory Collins, for the interview, and congratulations again on being this year's MRC Millennium Medal 2020 winner. So as I mentioned, Sir Rory will be presented with the medal and be given a virtual lecture on the evening of the 29th of March. So be sure to keep your eyes peeled for promotions.